Thank you very much. So I'm very honored to be here, and thanks for the invitation and the possibility to give this webinar. So what I want to do today, and as this says, as the title says, Harmony and Diversity, Exploring the Rich Microbiomes of South American Wildlife. So what I want to bring is some examples of a little bit of the research we have done in different South American animals and environments, and a little bit of what we have learned, and probably what we some of the take-home messages will be on some of the commonalities that we have found through all microbiomes, and what could even that say about our own microbiome. So I'm associate professor at Universidad de los Andes in Bogota, Colombia, South America, and I'm adjunct assistant professor at Washington University in St. Louis. So just to start, probably you're familiar about some of the countries that are these megadiversity hotspots, and it's been told that Colombia is a megadiverse country. And we always talk about the diversity of amphibians, diversity of plants, diversity of mammals, diversity of insects, diversity of plants. But we never talk much about the diversity of our microbiomes or the microbial biodiversity. So this is something I'm particularly interested in looking at. And the way we do it, and probably if you have attended some of the other webinars, you already know about this, is through metagenomics. That is the analysis of the genomic component of a microbial community, essentially selecting a given environment, extracting the whole community DNA to understand what is the community present and the structure of that community. And that's mainly what we do. And this is thanks to the increased capacity and decreasing sequencing cost of the sequencing capacity. So this slide is just showing a little bit of the increase in read length and gigabases per run with the different sequencer capacities. And we see up here most of the Illumina machines that are generating very high throughput sequencing, and some of the long reads around here, the MinI and the PacBio. So this is a little bit of what we used in our research. So just in general to describe, I'm the lead of the research group in microbial ecology and computational biology. And what we want to do is characterize microbial communities through metagenomics and use computational methods for the assembly and annotation of genomes. And I'm particularly interested in the study and characterization of viral communities. And together with that, we develop computational methods for the analysis and interpretation of omic data. So with that, we tend to work in many different environments. So we do a lot of viromes, that is those viral microbiomes. We do microbiomes, we do genomics, a lot of wet lab projects that are in the outer ring, a lot of dry lab projects or computational projects that are in the inner ring. And those constitute our main line of research. So today I'm just going to highlight a few of these microbiome studies that I would like to share with you. So to start, I wanted to start from smaller organisms going towards larger organisms. And I'm not going to bring today human data. I just wanted to go over precisely wildlife, so different animals, and at the end, we're going to see a couple of processes, but those occurring in non-experimental or laboratory conditions. So together with Elena, a former PhD student, she wanted to understand a little bit what was causing this necrotic patch disease in some corals in the Pacific. So we knew that this disease was quite prevalent, and you can see the symptoms here. Is patches that become necrotic in the coral, as the name suggests. So we wanted to see what the microbiomes would look like. So we wanted to take some samples in very close in proximity of the disease part, in the distant part of or basal part of the disease part, and then more or less within the same distance in a healthy colony. So we took some of those samples and we started looking at the microbiomes, and what we see in general is that when we have the healthy ones, just healthy in the left, you can see that it doesn't matter if it was proximal or basal, they all are spread and mixed. So there was no difference in their microbiomes. However, when we add the diseased ones, we can see that the basal part of the disease looked pretty much like a healthy microbiome, and only the proximal part is the one that started to become quite distant, and the microbiome seemed that it was changing significantly and diverging from the healthy ones. And we started characterizing what was in there, and we look at the healthy microbiome, and we can see that it's dominated by endozoicomonas and this mycoplasma, together with some other bacteria. So these are the common core bacteria present in the microbiomes of corals. Interestingly, here we were able to see that this mycoplasma was a bacteria that was relatively recently reported that was becoming to be highly abundant as part of the microbiome of the corals. And it seemed that it's displacing the natural endozoicomonas, which was the former most abundant bacteria. So you can see that we have three different OTUs of endozoicomonas. They were highly abundant in these samples, where the mycoplasma was low abundance. And then as the mycoplasma increased abundance, we have decreased abundance of the endozoicomonas. So you can even see there a little bit in the healthy ones of shifts in the composition of the microbiomes as this mycoplasma becomes more prevalent in the colonies or in the communities. But then when we added precisely the diseased ones, you can see the disease basal looks pretty much similar to the healthy ones, having a lot of endozoicomonas and the mycoplasma. But the proximal part, you can begin seeing some new bacteria appearing. Many of these are common members of the ocean waters, and some could be opportunistic pathogens. So we were not able to see any single bacteria that was exclusive from the disease part of it. So we don't think that necrotic patch disease is a bacterial caused disease. We think it will be more environmentally driven, but those changes in the environment are causing a change in the microbiome. And that change in the microbiome is reducing the normal microbiome that is present and allowing some environmental bacteria to come in. And that environmental bacteria could lead to potential pathogens. So from this study, so I want to bring in some take-home messages from each of the studies. So from here, just even from an organism as in quote unquote simple as corals, the microbiomes we can see already they're highly structured, and then disease conditions will alter them, and that's what we have been calling dysbiosis, and that allows environmental bacterial and potential pathogens to colonize. So that's a little bit of the main point of that early study. And then looking at other animals and now increasing a little bit of complexity, we're going to talk a little bit about bumblebees, and this was a work done with Nicole and was recently published. So we went through different places in Colombia, and in particular in Cundinamarca, and selected some different bumblebees from different species. And then we look at the microbiomes, and when we cluster those microbiomes, we saw that they pretty much cluster based on the species they belong to. So even in bumblebees, what this is suggesting that each species has their own microbiome, their own very specific microbiome, and these clusters even by subgenera. And then when we look a little bit more in detail, and probably these plots, you have already seen those, they're just clustering the samples by similarity of the community. We can see here a big shift or split around this line, where you can see most of the samples around here, and those samples are still clustered by species. And then on the left, you'll see a few other samples together with this in the corner, that these are thigators. So these thigators are not bumblebees, these are bees, but this has the particularity that these are solitary bees. So interestingly, the bumblebees are social bees, so they stay in the community. So being together in the community, that means that they share a lot of their microbiomes, that they have the possibility of passing the microbiomes from one to the other one. While the solitary bees, they don't have that much opportunity. And when we look at the composition of those microbiomes and we try to align precisely this dotted line with the bottom part, suggesting that all these in the left are essentially all these that we can see here. And we can see that they mainly have that corbiculate core microbiome. So it is the core that has been reported for all corbiculate insects, in particular bees and bumblebees. So, and then on the right, what we can see is a very different microbiome, mostly dominated by environmental bacteria. And this again is what is dominating mainly those solitary bees. So what we can see here is that similar to what was happening with the corals, when there is a disruption, here is not necessarily a disease, or it's not known to be a disease, that will shift the microbiomes and reduce the regular corbiculate core microbiomes and increase a lot of the environmental bacteria. Now we have a lot of environmental bacterial potential pathogens in there, and they have a disturbed microbiome, which probably that will cause different health problems with the bumblebees. We also sample other bees, or compare the data with other bumblebees that we have. So all these bees were collected, or bumblebees were collected from Colombia in the neotropics. And it's the first time we were characterizing the microbiomes of the neotropical bumblebees. And there were a lot of data from temperate bumblebees. And when we make a phylogenetic tree based on the structure of the community, what we can see is that it's clusters very strongly with the subgenus, except for all the disturbed ones, all these ones that are here on the right, together with some disturbed ones that we find from temperate regions and the other species. So all disturbed ones seems to cluster together based on this composition of mainly environmental bacteria, and all the other subgenus, they will cluster together by subgenus. So again, there is the message here, and I'll be showing very short examples of the one I just showed, is that the microbiomes has very strong coevolution with the host. You can see that we can detect very strong signal based on the microbiomes associated with the subgenus of the insect. And we have conservation even across great distances, having neotropical and temperate bumblebees having the same type of bacteria and the same type of microbial structure. But they are very fragile to changing environmental conditions. So whenever there's a disease or environmental condition is changing, we'll see this dysbiosis reducing the normal community and allowing environmental bacteria to colonize. So this is seen with the bumblebees. Now, I'm going to shift to a different organisms, and now I wanted to talk a little bit about shrimp. So together with Leda Restrepo, we were doing this in collaboration with the Centro Nacional de Acuacultura e Investigaciones Marinas in Ecuador. So this is the shrimp industry in Ecuador. And we wanted to characterize some probiotic species and their effect that they have in the microbiome upon disease. So I'm going to show briefly this experimental design. So we'll have different pools of, this would be the control, just healthy shrimp. Then we'll have these healthy shrimp together with a Vibrio bacteria. This is the strain name, and this Vibrio bacteria is known to cause a disease known as HPND. AHPND. This disease is very important for the shrimp industry and has caused massive losses in the industry. Then the other two branches of the experiment was having the shrimp with the pathogen and this illus strain that was one of the probiotics that we wanted to test. And the other one will be the same, the healthy shrimp, the pathogen, and another probiotic that was a bacillus strain. So we have healthy shrimp with the healthy microbiome, with a pathogen, and with a probiotic, two different types of probiotics. We fed the mice the probiotic for a month prior to the challenge, then we challenge with the pathogen, and we see what happens. So the one that had just the pathogen is here in blue. So you can see mortality appearing up almost 100% mortality just within the first 30 hours. So this is how aggressive this pathogen is. It will reach 100% mortality really fast, even in industrial scale. Then the next one we'll have is the Bacillus probiotic. We can see that there's a delay in the mortality. We don't see mortality right from the beginning. It's a little bit delayed, but eventually it will reach almost 100%, not 100%, but almost 100%. And then the ELI probiotic not only has further delayed mortality, but it never reached 100%, suggesting that the ELI probiotic is doing a good job in protecting the shrimp from the virulence of the pathogen. And in our negative control, we have no mortality at all. So the first thing we wanted to check was against this alpha diversity, because as I've shown you before, whenever we have these alterations of the microbiomes due to pathogens or a disease or environmental conditions, one clear signal usually is the decrease in microbial diversity. So we can see here the Shannon diversity of the healthy shrimp, and then in the diseased one, we can see a significant drop in that alpha diversity. Interestingly, the ELI probiotic prevents that drop in alpha diversity and maintains that alpha diversity in levels equivalent to the healthy one. While the Bacillus probiotic, that was the one that was not able to prevent the mortality, was not able to prevent that loss of microbial diversity. So now it's suggesting that it is doing the fact of adding a probiotic is having an effect in the microbial community overall. And when we look at the beta diversity, how the microbial community is clustering in the pathopancreas, that is an organ that is at the end affected by the Vibrio infection, we don't see much of a pattern. But in the stomach, that is where early onset of the disease will happen, we can see that the infected shrimp are on this side together with the Bacillus probiotic, and the healthy shrimp are on this side together with the ELI probiotic. So it is appearing that it not only helps maintaining a high diversity, but that the structure of the community itself is not changing significantly. So this ELI probiotic is helping the bacterial diversity to be maintained. And precisely, we are able to look for potential markers, or in this case, they're amplicon sequence biomes, which correspond to taxons. However, these were not assigned to any species or genus level taxon. But what we can see from all these 4 taxons is that we have in general a high abundance of them in the healthy shrimp. In the diseased shrimp, we don't usually see those bacteria. And interestingly, the ELI probiotic is able to maintain the high abundance of those healthy markers, while the Bacillus probiotic is not. So we can see that in this ASB, the same in this one, very marked in this one, and in this one as well. I have to mention that all these 4 ASBs were not selected based on the abundance in either probiotic. We just use the abundance in the healthy and the disease to identify the healthy markers. So no data was used at that point about the different probiotics. So just to point out something that is particularly interesting from this is that this ELI strain is actually a Vibrio strain. It's a Vibrio that is taxonomically very closely related to the pathogen that is causing the disease, while the Bacillus is taxonomically very distant. And this could be very important because from this study, what I want you to take home is that the microbiomes can be protective of potential pathogens. So our microbiomes is our very first line of defense against any pathogen that is coming in, against any bacteria that is coming in. The first thing that they will encounter when they reach us is our microbiome. So if we have a strong microbiome, that will be protective. In particular, if the microbiome we have display a similar niche. And that's what we think was happening with the Bacillus and the ELI strain. So when the Vibrio is arriving, the Bacillus strain is not a good competitor because it's not taxonomically similar. It doesn't have similar functions, so it's not able to compete against that Vibrio strain, and that's why the disease will progress. However, if we have a Vibrio already in place, and a Vibrio that does not cause any disease, as this Illy strain, that is a good competitor that when the other pathogenic Vibrio will arrive, it will prevent that Vibrio from colonizing and overall preventing the disease development. So this is an important information about our microbiome. Having a diverse microbiome, having microbiomes that may be able to compete, or bacteria that may be able to compete with potential pathogens, constitute a very important first line of defense against any potential disease. And now I'm going to go bigger to mammals, to bears. And this was a study done mainly by Andrea Borpon and about the gut microbiota in Andean bears. So Andean bear for Latin America, it's very important because it's the only bear species that we can find in South America. It has this very wide altitudinal distribution, and also it will go from lower altitudes to high up in the Andean mountains. This Andean bear is considered vulnerable, it's still not considered endangered, but it's in its way towards there. And it's been mainly because of habitat fragmentation and degradation. As we begin building more roads and we have more farming towards the regular ecosystems that the bear used to inhabit, there is less and less of the habitat of the bear. So this is what is bringing it from vulnerable to endangered. And also, of course, negative interactions with humans. In particular, there are a lot of farmers that claim that the bear may eat their cattle, and because of that, they will hunt the bears. So this has put this bear into particular attention for their vulnerability. But importantly, together with like the panda bear, this bear is mainly herbivore. So even though most of the bears are carnivores, and most of the bears will have a carnivore-like gastrointestinal tract, and most of the bears will have a carnivore microbiome, both the Andean bear and the pandas, because of displacement of niche precisely, they had to switch their diet to herbivore mainly diet. So these are the type of plants they consume. They have predominantly plant-based diet. They may consume some insects and even some small animals, but that is usually incidental and not intentional. And there is no physical adaptation for herbivory. So these animals have the carnivores like gastrointestinal tract. The fact that they don't have herbivory type gastrointestinal tract means that they don't have an organ like the rumen or something similar that will allow for the fermentation and degradation of plant-based fibers. So they only have the carnivore GI tract. So they rely completely on the microbiome in order to be able to degrade some of that plant. As I mentioned, as there's habitat fragmentation, this means changing in the type of plants that we have available and will change what is available for the bear. Also, many of the, or some of the bears have been captured and they are kept in captivity. And the captivity diet, it is around 3,700 calories per day, kilocalories per day, and is supplemented primarily with domesticated fruits and cereals. So it's very, very different from the diet that they will have in the wild. So what we wanted to see was what happened when there's a diet switch going from the wild diet to captivity diet, and if this diet may affect their microbiome. And given that the bear is completely reliant on the microbiome to be able to extract the nutrients from the food, this will have a direct implication on the bear's health. So what we did was, again, in Cundinamarca, in a given place in the Andes. We went there and collect several samples from wild bears, together with some samples from a zoo that have been permanently captive for many years now. And we had the opportunity to sample 2 bear cubs that, although they were born in the wild, they were captured really at a really young age, and they were maintained in captivity in a center with the aim of reintroducing them later on to the wild. So the first thing we can see again when we look at alpha diversity is that the highest diversity we can observe is coming from the wild bears, as we will expect. Then those cubs that were wild and then they were caught, there are intermediate diversity, and those that have been in permanent captivity has the lowest diversity of them all. So again, as we have shown with the other animals, when you modify the environment, modify in this case the diet, there will be an important loss of diversity, and this will be analogous of some disease, or this will be causing that dysbiosis. When we cluster and we do the beta diversity, you will see all the wild samples up here, while all of the permanent captivity and most of the wild caught right here. You may see a wild caught sample right up here with the wild samples, and here in the tree, like there's 2 samples. Those correspond to the very first time point of those wild-caught bears when they still had some of those wild-type microbiome present. When we look at the microbiome composition, and this is at the phylum level, I'm not going to go in detail, but probably will be pretty obvious that if you see this part where the wild samples are, compared with the permanent captivity in particular, you can see a big change, a big drop in the Bacteroidetes, a big drop in these proteobacteria, and a high increase of many firmicutes. So this is what is changing in many of those permanent captive bears. When we try to see what's the overlap between the different animals, we can see that they still have a significant overlap in the bacteria that we find on the 3 of them, which is encouraging. However, there was a good number of strains that are present only on the wild. We never saw those bacteria in either the permanent captivity or the wild-born or there are some shared, definitely more shared with the wild-caught bears, and there's almost anything that is unique for the permanent captivity or the wild-caught, and there's only one that is common between the 2 of those. This again brings a point that what it's happening is not that captivity is bringing new bacteria because we're bringing new diet. What is happening is that they are just losing bacteria, losing diversity, and losing capacity to degrade different plant material. So this what shows us, and a little bit of what I wanted to show here is that here we have from the cubs microbiome time of sampling, and what we see is distances to the captive, and we can see that the early time points were more or less distant to the captive microbiome, and as they remain captive, the microbiome begins looking more and more and more like the captive one. And this is a different way of looking at the same data, and the number of shared bacteria with the wild individuals. So as they look more like the captive ones, they are sharing less and less bacteria with the wild individuals. So both of these plots are showing that having these cubs in captivity is damaging their microbiomes. And if we are planning to bring them back or reintroduce them to the wild, the question that remains is like, have we caused so much damage that this microbiome will not be able to recover, and then the bear will be at a disadvantage because the bear will not be able to process the type of food that will eat in the wild, and that will potentially cause significant disease problems or just a significant malnutrition in the bears. So that's something that we are trying to tell the people that keep the bears both in the zoos and these reintroduction centers to help them bring more of the native diet so that there's not such a big loss of their microbiomes. So the takeaway here is that the structure of the microbiome can be highly influenced by diet and may cause potential permanent changes in the microbiome. So when you change your diet and you keep a restricted diet, what you're causing is to lose many important members of the microbiome. And this is relevant not only to bears, but probably to any animal in general. And we wanted to follow this up with a different setup, and now we're going to look at different animals. So this is woolly monkeys, and this was a work led mainly by Camilo Quiroga and Luis Chica in the lab. So what they were doing was following these woolly monkeys during a reintroduction process. So what we had here was it was a very limited data set. It was just 3 animals, but they have been kept in captivity for a while, and then they were being reintroduced to the wild. And it was interesting because during this reintroduction period, the animals they also lose the capacity to find their own food, because in captivity they are just use of the food being provided to them, while in the wild they have to go and look for the food. So at the beginning there was a transition period where some of the food was the food that they will eat in captivity will be provided to them as well, just so if they wanted to come and take it, they will be able to take it. So if we look at the Shannon index, as we expected, the diversity in the captive bears and the captive monkeys, as happened with the bears, was lower than once they were released. So diversity increases when you were released and you went back to the wild diet. Interestingly, when we look at the diet diversity, because this was also evaluated, in captivity there was less diet diversity than when they were released. And actually we can see that in different time points where we have groups where we had the microbiomes analyzed before they were released, at the beginning after they were released, and then one of the individuals. So we have 3 individuals. One, unfortunately, we're not able to sample it anymore. It was gone from and unavailable. The other 2, one remained in the wild, and the other one went back, and it was captured back again. It didn't, and it does happen with certain frequency that the animals just don't get accommodated to the wild setting again, and they rather come back to where they were kept before. So that happened with one animal as well. And what we see is when that animal come back, again, the diet diversity and the microbial diversity will come back down. And if we look now again, beta diversity, we have all 3 animals here, and you can see those by different colors. And you can see by the direction of the arrow how the microbiomes were. So most of them will start around here in the center, and you will see they start here with more or less a common microbiome in captivity, and this one will just try to go far apart as they go into the release part. The same with this one, it will just in captivity, and now it's released, so it's go farther apart. And this was the animal that started in captivity. It went to release, and then it came back and was captive again. So it's very interesting to see how the captivity does make a more homogeneous, less diverse microbiome. And when you go out in the wild, you increase that diversity, you begin getting new bacteria. So this was a very important point for us in terms of that microbial diversity, because with the bears, we had the question, what will happen when we reintroduce them? Will they be able to gain back their bacteria? And even though we don't know that answer with the bears, with the woolly monkeys, apparently as soon as they begin eating back again the wild diet, they will regain that wild microbiomes back again, and they will increase their microbial diversity. So there is hope that when they start eating the regular diet, the microbes will come back again. So from here, in comparison with the bird, the idea is that bringing back the right diet may bring back the good microbes. So in general, this has been also seen with the human microbiome, with a Western diet losing diversity, losing the microbiomes, and then when going back and bringing back the right diet will bring back some of those good microbes, as long as it is done on time and before definitely losing some of the microbes. So now I'm going to shift a little bit and not talk about completely wild animals, but these are a couple of industrial processes and that also require microbial communities. And this was a study led by Mauricio Pacheco, where we wanted to look at the fermentation of cacao. So probably you all like chocolate, probably you have tried chocolate in many different ways. And chocolate comes from this plant that is the cacao. This is the cacao fruit. And when you open the cacao fruit, it will look like this. And this is a layer of almost a mucus layer, it's a mucilage. And this needs to be degraded and fermented in order to provide the seeds with the right flavors for the production of chocolate. So before it was used very in a handcraft type of way. So it's very particular to each region and each producer with these wooden boxes, and they talk about this as a drying process. But what we actually know now is that this is a fermenting process where a microbial community has to play a very important role in it and changing a lot of the different metabolite aspects of the fruit in order to give the right flavor. So what we did here was in different regions of Colombia, we selected 3 different farms. I'm going to just show the data of 2 of them. And what we did was follow in dry season and wet season or our whole production period for 144 hours with sampling every 12 hours in 2 different farms. So interestingly, what we saw, we follow bacterial and yeast abundance, is that it's very low diversity and very consistent succession events. So we start with Enterobacteriaceae that will eat those simple, and yeast that will eat those simple sugars and generate alcohols that then the lactic acid bacteria will take those alcohols and produce lactic acid that will begin decreasing the pH, and then will come these acetic acid bacteria that will be able to thrive in these acidic conditions and generate acetic acid as well. So this succession is very consistent, in different times, is very consistent in different zones of the box. You may see a little disturbance here, and the problem here was that early on, the farmer decided to mix the box, and that mixing implied entering oxygen into the box, and that oxygen changed the dynamics and the succession of the microbial diversity, seemingly disturbing the process, even though later on it will go back to what will be expected, but it does change some of the organoleptic properties of the chocolate. And even though this will seem like a very simple process with essentially 4 or 5 different bacteria participating, when we look in more detail, almost at the strain level, what you will see here, so this is one farm, here's the other farm, dry season, wet season, upper zone of the box, lower zone of the box, and this is the Enterobacteriaceae, this is the lactic acid bacteria, this is the acetic bacteria, and what you see here is that early on, you have a very broad collection of different enteric bacteria. that with time they will be selected to be at the end only one. And then the same thing will happen when lactic acid bacteria will appear. You will have a big diversity of those, selecting for mainly one of them. And then when the acetic acid bacteria, you also have a big diversity, and over time selecting for just one strain. So now, again, and similarly to what happened with the Vibrio, is that when you need a particular functional type, you may start with a big diversity of those bacteria, but then they will be competing against each other, rapidly selecting for just one. Even though overall the community, you will see similar diversity. When you look at genus or even broader taxonomical level, the community will seem very stable or with the expected successions. But at the strain level, there is a lot of competition going on and selection for the fittest going on in the microbiome. So in this fermentation process, like other microbial process, there's an ordered, predictable succession of microbes based on the functional abilities and requirements. And finally, this example led by Maria Antonia Madrid was in the coffee fermentation, because similar to chocolate fermentation, coffee also needs to have a fermentation with a microbial community in order to get the flavors. So we tried to look at an inoculated with a yeast started culture for 100 hours fermentation process and a regular fermentation process. We took the green coffee beans, then they were toasted, and then we prepared cups that then when we have three standard cupping, fermentation cupping to test the different coffees, and they were able to determine that the inoculated have a better sensorial profile than the non-inoculated coffee. So we wanted to see what was making this inoculated coffee better based on these tasting. And we also did, in this case, metabolomics on both the green and the roasted coffee. So in terms of bacterial diversity, we look at the bacterial taxonomy and the fungal taxonomy, both in the inoculated and the standard. And what we see is that early on both, we have a large diversity, both in the inoculated and the standard, but then is really rapidly selected in a very similar way to what happened with the chocolate, where we have selection of enteric bacteria, then lactic acid bacteria, and very little of acetic acid bacteria. If you see, this process is just for 100 hours, while chocolate is 144 hours. That allows for more of the acetic bacteria to increase in abundance. Here with the inoculated, with the coffee, we don't want the fermentation to go that far because the acetic acid will provide some organoleptic properties that are not desirable, in particular in the case of coffee. So what in general we see is that what the inoculation helps is stabilizing the microbiome, in particular in the fungal part. We see that after inoculation, there is a stabilization very fast of the microbiome, while in the non-inoculated coffee, you see a more diverse and less stable microbiome happening. But more than this stability in the microbiome, what happened with the metabolic profile? So we tested a bunch of different metabolites, both in the inoculated and the standard coffee, both in the green and the roasted coffee. And we can see that even though that the green coffee, as the cuppers tasted, had more bitter taste and had also a little bit more sweet and had some more caramel notes. While the inoculated coffee was a little better in terms of this was in the green coffee, but has more fruital notes and has a lot of more of the innate coffee metabolites that helps in determining the coffee quality. So currently we're trying to associate which microbes are involved in the generation of these metabolites, but it's interesting that we're capable of modifying a fermentation process with this started culture, that we can trace that into modified metabolite profile, and at the end we can trace that to a better flavor in the tasting. And with that, the final product of a microbial process highly influences the properties of the product, as we saw in this coffee fermentation. And I hope that with this, I have shown you different examples in very different environments and different model organisms, but all those bring these take-home messages about the importance of our microbiome, having our microbiome diverse, how the microbiome responds on disease, how the microbiome responds in different diets, and how the microbiomes in fermenting process have very structured, predictable successions that are completely and intimately linked to the consequences, in this case, the flavor of the food. But in our case, it will be completely associated with our capacity to assimilate different foods and our health. And with this, I want to thank you. Thank you for your attention. And I want to thank mainly all the great people that I just show you a handful of examples of the type of things we have done. But many, many people have gone over the labs over the years. And with this, I will leave you and I'll be happy to take any questions.
Thank you. Thanks a lot, Alejandro, for a wonderful presentation. We already have quite a few questions in Q&A box, and we still have about 10 minutes to answer them. So I'll read out the question and you can answer them. And yeah, I would also like to thank everyone for joining the webinar today and sending in your questions. So first question is about, I think, one of the starting slides where you mentioned about ASV and OTU. So the question is, ASV are currently in use instead of OTUs. Can you comment on that?
Definitely. So the OTU is a term for operative taxonomical unit that comes from a traditional macroecology, and what it is meant to be is it should be a proxy to species level. And when many of these meta-taxonomy studies started with amplicon-based sequence, there was the assumption that clustering those amplicons at 97% identity will be the good proxy for species level bacteria. So that's why at the beginning, many of these studies will cluster at 97% identity and call that an OTU. However, with time, we have seen that that cutoff is very artificial and it does not correspond to species level. So nowadays, what they do is that, and the other reason why 97% was used was in order to be a little robust to potential sequencing errors. So now that the technologies are getting better and we have better software in detecting potential sequencing errors, what we do is correct for those sequencing errors and then just cluster everything at 100% identity. So that may be, and that's why it's called amplicon sequence variant, because they're 100% identical. Anything that's 100% identical is being clustered together. And they may not reflect necessarily species. They may be strains. You may have more than one ASV that correspond to the same species. So in order to differentiate that different processing of what is being doing, that's why they're being called ASVs and not OTUs anymore.
Okay, thank you. The next one is, thanks for the interesting session. How does a change in diet affect the gut microbiome? And how stable is this change? And what does it take to disrupt it?
Not only based on what we have seen in animals, but of a lot of what we have seen in humans as well. Diet is the most important factor affecting the composition of a microbiome. With that, I need to make probably a distinction in which the structure of your microbiome, meaning who is there, that is mainly determined at very young age. So for us humans, mostly at age 3, we already have the bacteria that are going to be with us for the rest of our lives. It's really hard after the age of 3 to get colonized by new bacteria. It's not that difficult to remove some of that bacteria. We don't provide with the right diet. But what will happen is that the bacteria that we have is hopefully a highly diverse community capable of degrading most of the dietary foods that we take. And their abundances will really rapidly change with our diet. So within 12 hours, 24 hours, we can have a complete shift on microbiome. So if for today, I have for lunch and dinner a big salad with greens and many different vegetables, all the bacteria that are capable of eating those vegetables and those fibers will flourish. And if by tomorrow I just eat carbs and hamburgers and fats, all that bacteria that are raised, they'll be gone to very, very low levels of abundance. Almost many times almost undetectable by our common methods. But they're not completely lost. They're still there. So that's why the idea is keep varying your diet, keep giving healthy diet, and keep varying the different components of that healthy diet. Include plant fiber every single day, ideally in different methods to keep as diverse as possible microbiome.
Thank you. That's very interesting. So next one is very interesting session. I have a question about organisms living in symbiotic relationship. What is expected of their microbiome composition if they are living in similar environment? Should we expect similar microbiome for both organisms?
Not necessarily. Environment is a factor, as I mentioned. More important than environment, we have two factors, that is genetics and diet. So two organisms may be living in the same environment, but if they don't have the same diet, they will not have the same microbiome. And even if they have the same diet, it will depend a lot on the genetics itself of the organism. As I mentioned with the bumblebees, we have all these bumblebees were collected essentially in the same environment. All those bumblebees were eating essentially the same type of pollen from the plants, but we saw a very strong phylogenetic signal. So in particular, the bacterial species that will be colonizing, and we have seen that the bacterial species that you'll find in humans are really closely related, but not the same ones as the bacterial species you'll find in chimpanzees or other monkeys. So the phylogenetic relationship is quite strong and important, and there's been this very, very strong coevolution with our microbiome. That means that in terms of who is there, is determined pretty much by our genetics. In terms of how much of each bacteria is present, it will be driven mainly by the diet that is intrinsically related with the environment where they are.
Okay, thank you. What is the relevance of longitudinal studies that assesses effects of probiotic therapy, for instance, by the example of bumblebees and beavers? If it is relevant, what are the challenges, specifically studies that span generations of host organisms?
There'll be different factors there because you will be introducing a species like a probiotic, and the question is for how long are you going to be introducing that species? So the one I showed with the shrimps, we were giving the probiotic for a month and then just testing the capacity of that bacteria to protect the shrimp for the next few hours. In humans, it depends also on what probiotic you give. So, for example, the human probiotics that we usually consume that are lactobacilli, those are not our native bacteria. And as I mentioned, there's such a strong relationship with our native bacteria that those probiotics will not usually colonize. And that's why every time we consume probiotics or every time we have the yogurts, they say consume daily for a better effect because those bacteria don't usually colonize. So as at the moment you stop providing the probiotic, the bacteria is going to be gone. Now there's research on better probiotics closer in relationship to the actual bacteria that colonize our guts. And those probiotics are expected that if you give the probiotic together with the right diet, again, the importance of the diet, the probiotic may engraft and will stay within the microbiome for as long as the right dietary components for that probiotic is given. So it is challenging sometimes to do long-term longitudinal studies, probably with model organisms like bumblebees or something that is shorter lived. It will be easier to follow them and see the effect. With more complex organisms such as mammals or longer lived animals, will be more challenging and costly, but is doable. But I don't think they need to be done for that long and even for generations. Usually, when a bacteria engraft, and the diet is given, what we have seen is that if it's engrafted in the parents, it will be transmitted to the next generation.
Okay, thank you. Could you suggest some of the softwares that you used for the analysis?
In terms of software, for most of these studies that I showed is amplicon-based sequencing. So it's just amplifying the 16S region or the ITS region. We use QIIME2 for most of them, but probably like the bear and the cacao one that I think are the oldest ones. Those we use QIIME1. But currently we're using QIIME2 and the coral is also a relatively old one. For all the other ones, we're using QIIME2 all the way to generating ASV tables. And once we have ASV tables, we use different libraries in the R software, the statistical software, to do most of the statistical analysis. Then just generating the figures and so on is a little combination of R, QIIME, and a little bit of editing with Illustrator to just improve visibility and so on.
Thank you. So it's now the full hour, but we can take probably one last question, Alejandro, if you don't mind.
Sure.
So is it possible to introduce a specific flavor by just using a certain inoculum, irrespective of the type of coffee beans used?
Yes, and that's part of what is being tried. So right now, you may see that if you go to a Starbucks, just to say something, there's vanilla coffee and other type of coffee flavor. Those flavors are added afterwards. Those flavors are added even after roasting, and they're completely artificial. What many of the farmers are trying is that by adding starter cultures, give the fermentation process allow the fermentation process to generate the specific flavor you want. So we know that addition of certain yeast and certain bacteria will generate certain organoleptic profiles, giving different flavors to the coffees. So now that's a more natural way of doing it to make the coffee generate that desired flavors since the start, and not adding that chemically at the end. Oh, thank you all. Have a great day.